is a procedure used to treat genetic problems by substituting healthy genes for faulty ones. DNA is inserted into human body cells via an artificial technique. In 1989, the first successful gene therapy procedure was completed. In this figure, a normal gene is injected into the cell that has a faulty gene to help the cell function normally. This method is primarily used to treat genetic disorders and battle diseases that affect the human body. DNA is inserted into the cell to replace the damaged proteins in that cell. In most cases, disorders result from the cell's incorrect production of proteins. Gene therapy is a method used to treat these diseases. For instance, cancer cells have defective cells that are distinct from healthy ones and have malfunctioning proteins. Therefore, this disease would prove to be fatal if these proteins were not replaced. There are two types of gene therapy, somatic and germline gene therapy. Somatic gene therapy typically takes place in the body's somatic cells. This pertains to a single individual, and the damaged cells will only be replaced in that individual by healthy ones. This technique involves transferring therapeutic genes into the body's stem cells or somatic cells. This approach to gene therapy is regarded as the most effective and secure. However, germline gene therapy occurs in the germline cells of the body. This approach is typically used to treat genetic variations of genes that cause disease and are inherited by a child's parents. The procedure is adding a healthy DNA strand to the cells that make sperm, eggs, or other reproductive cells. Since the dangers of germline gene therapy outweigh the benefits, it is illegal in many states.
The approaches to gene therapy. Gene replacement therapy. Gene replacement therapy involves replacement of a mutated or defective gene with a functional gene. This approach is suitable for genetic disorders that result from a single gene defect, such as cystic fibrosis and sickle cell anemia. Number 2 is gene editing therapy. Gene editing therapy involves the direct alteration of DNA sequence in a patient's cells to correct or eliminate genetic defects. This approach is used to treat genetic disorders caused by mutations or deletion in the DNA sequence. Number 3 is gene silencing therapy. Gene silencing therapy involves the use of small interfering RNA molecules to reduce or eliminate the expression of specific genes. This approach is used to treat diseases caused by the overexpression of genes such as cancer and HIV. 4. Immunotherapy. Immunotherapy involves the use of genetically modified cells to target and destroy cancer cells. This approach uses immune cells that are modified to recognize and attack cancer cells specifically. Number 5 is gene augmentation therapy. Gene augmentation therapy involves the addition of new genes to cells to compensate for the defective genes. This approach is used to treat genetic diseases such as muscular dystrophy and hemophilia.
Dr. Theodore Friedman is a respected endocrinologist at Charles R. University, specializing in hormone-related disorders, pituitary, thyroid, adrenal, Cushing's disease. Board certified in internal medicine, endocrinology, diabetes, and metabolism. He is also a professor in cardiometabolic diseases and actively involved in clinical research. Richard Robin is a Canadian contemporary abstract artist, has exhibited his work globally, including North America, Europe, the Orient, and India. He is a master of Canadian abstract art, known for his luminous clarity inspired by the prairie sky.